right, we are recording, but there's about 10 minutes left, so feel free to skip ahead about 9 minutes, 10 minutes. Before you do, though, I'll go ahead and tell you how the instructions for today are pretty much the same. If you're watching this video, you need to write down and send the attendance words. But in addition to doing that, you need to take a screenshot when I say the words. So you're going to be sending a list of words and a list of screenshots. And at this point, now you can go ahead and skip ahead about 10 minutes if you want. Morning. Morning. All right, there's issues with the screen. So I can't have one screen here and one screen there. Therefore, I cannot do attendance the way I usually do, because usually I have it pulled up here, because that's your attendance grade, and I put that in there. I can't do that now, because then it would show everybody's attendance, and that's against the rules. So for today's attendance, even if you're in person, you're gonna need to send me the words. Does that make sense? Okay. It's a pain, but I don't know if people mess with this, it pisses me off. And it is 8 o'clock, so we'll get started. Again, like I said, even if you're in person, you need to send me the words today, because I can't pull up my spreadsheet for attendance. If you're online, it's the normal thing. You need to send me the words before 9 a.m. And at the end, including you guys in person, your last word for attendance is going to be send me a question about the content that I cover. So it's going to be business as usual for the most part. If you're watching the video, so not you guys who are online right now, but if you're watching the video, if you've gone to YouTube and you're watching this video at a later date, in addition to sending the words, you also need to send a screenshot of when I said the words. So that's the announcement. Well, that's the attendance stuff. We'll do the announcements after a few minutes as usual, give some time for the late people to get in here. And here's where we left off. Right, do you have any questions before we get started? Back into it? All right, so we left off talking about the sales cycle. We've already talked about this slide right here. This is old news. I left off showing you this, and I was saying that the number one thing you should memorize first is the fact that the entire sales cycle is broken down into interphase and the mitotic phase. Memorize that before you memorize all the other stuff. Somebody had the wrong answer, but I love this wrong answer, because at least you were on the right page and it shows you're probably paying at least some sort of attention. One of the last questions for attendance last Wednesday was, what the sales cycle is broken down to 2 phases. blank phase and the mitotic phase. And someone wrote S phase, so I like that because I don't even think I said the word S phase. I may have, but anyway, it was up there, so at least they were looking at the screen. But yes, cell cycle itself, the big whole cell cycle is broken down into two phases: interphase and mitotic phase. Now you can see here, interphase is broken down into different phases, and or stages and phases, and we'll talk about that. We'll also talk later about how mitotic phase is also broken down into different stages. And you will need to know the difference between these things. So let's get started. This is the new stuff here. Again, the cell cycle is broken down into two stages: interphase and mitotic phase. But here's some new information that you do need to know. The cell is almost always in interphase. At least 90% of the cell cycle is made up by the interphase. You don't necessarily need to know that number, but if there's a question that says, the cell is usually in what? Mitotic phase or interphase? The answer is interphase. This is when the cell is just performing its normal functions. Depending on what kind of cell it is, different cells have different jobs, but whatever its job is, that's what it's doing during interphase. There's also a part during interphase, so not the whole thing. So I'm going to put like a little star next to this. So it's not during the whole interphase that this happens, but a specific part of the interphase, it doubles everything in its cytoplasm, right? Because it's getting ready to split, eventually will split. So that's one of the things it has to do to get ready. And it also grows in size. So the first word for attendance is, pay attention here. The cell cycle is broken down into interphase and the mitotic phase. Now the cell is usually in which phase? Interphase or the mitotic phase? Most of the time, the cell is in which? Interphase or mitotic phase? Any questions about this slide before we move forward? All right. So here we go. Now we're going to break down interphase and talk about the different phases in it. I'll go ahead and tell you this. I'm probably not going to ask you any questions about the different parts of interphase. There is one that's going to be a little bit more important, and you'll need to know about that one. I'll tell you when we get there. G1, I'm probably not going to ask any questions about, but you do need to know this. Before I start talking about this, the way it goes is first we have G1, then we have S, then we have G2. Those are the three parts of interphase. If anything, I might ask you to know that in order. G1, then S phase, then G2. But again, I'm almost definitely not going to ask you questions about G1 or G2, but I will ask you questions about the middle, which is the S phase. Because as far as we're concerned, in this discussion, S phase is more important, and you'll see why. Anyway, again, G1 is the first part of interphase. It is before the S phase. Obviously, again, like I was saying, you kind of need to know the order, even if you don't memorize what happens in G1. You do need to know that it is the first part, and then it comes before the S phase. At this point, the chromosomes they're just single, right? They haven't duplicated yet. So for humans anyway, each of the 23 pairs of chromosomes exists as a single entity. During this time, the cells are just performing the normal functions, but they are also growing, right? Because again, remember we're talking the cell cycle. So since this is the first part of the cell cycle, G1, this is like the birth of the cell, if you will, because at the end of the last cell cycle, there was one cell and it split into two new cells. Well, this is the beginning of one of those two new cells, right? This is where it's birth, if you want to call it that. It's not really a birth, but this is where it first came into existence. So it's at its smallest, and it's going to grow. It's going to accumulate energy, like in the form of ATP, for example. It's going to accumulate matter, right? All these biological molecules that you learned about earlier in the chapter. I will put really quickly before we move forward a big old X through here indicating, and I'm probably not going to ask questions about the specifics of G1. Probably. Anyway, any questions about G1? All right, let's talk about the one that's relatively important for us, the S phase. For this chapter, this is an important phase for us. They're all equally important for life, but for our discussion about cellular reproduction, the S phase is more important than the other parts of interphase, because this is basically when it's starting the process of getting ready to duplicate, right? So again, S phase is part of interphase. Specifically, as you need to know for the exam, it is the second part of interphase, right? Because it comes after G1, but before G2. Regarding cellular reproduction, like I've already said when I first pulled this slide up. It is the most important phase of interphase. Again, for this conversation, since we're focused on how cells reproduce, this is the most important part of interphase. This is when the centrosome is duplicated. I wish I could remember why I had that little star there. I would look if I had a regular screen, but my screen's messed up. But yeah, the centrosome is duplicated. I will definitely go ahead and put an X through that. I won't ask you about that. But more importantly, and as far as our conversation, this is probably the most important bullet point. This is when chromosome duplication happens. That will definitely be a test question. In which phase does chromosome duplication occur? And the answer is S phase of interphase, or maybe just S phase. So this is that stuff we were talking about on Wednesday when I showed you that picture of the singular chromosome, and then it duplicated, so then you had two sister chromatids that were attached together at the centromere. That is what this is, right? So that thing that I showed you days ago, actually, I'll just go ahead and show you really quickly and come back to it. This picture, right? This is what it looks like in G1. And then during S phase, this happens, right? During S phase, we go from the single chromosome, and then it duplicates into those two sister chromatids that are attached to the centromere that is not shown in that picture. And you definitely need to know that happens. And the way you can remember this, I hope, so this little last bullet point is like a little hint to help you remember this. S, you can think of S as standing for synthesis, right? Specifically DNA synthesis, because this is where we duplicated the chromosomes. Remember, the chromosomes are, as far as we're concerned, I mean, yeah, technically it's half protein and half DNA, but it's the DNA we're concerned with mostly. So any questions about the S phase? Some good independent work topics you can look into if you want. I just thought of this because I haven't mentioned this and you don't need to know it, but I just told you earlier that the entire interphase, the cell is usually about 90% of the cell cycle, right? So how much of the cell cycle is G1? Like what percentage is G1? What percentage is the S phase? Not that you need to know that, but again, it's a good independent work topic. And some of you guys really need to get started. It's 100 points, it's an entire letter grade. So for some of you, if you've gotten really bad exam scores or really bad lab scores, that independent work might be your key to bump you up to a D. You know, it might save you. Anyway, G2. G2 stands for the gap, you know, the second gap, now that you need to know that. Again, I'm probably not going to ask you the questions about this other than the fact that you need to know it is part of interphase. It comes after the S phase, and it's the last part, really. I should write that too. It's the last part of interphase. This is the last part before we leave interphase and go into this mitotic phase that we're going to talk about later. At this point, each chromosome consists of two sister chromatids, but you already knew that, right? So even though I'm not going to ask you that little bullet point right here, I'm not going to ask you that in an exam, you should still know it, because I will ask what happened in S phase. And remember, in S phase, we went from one chromosome to duplicated chromosomes, right? So two sister chromatids. So logically, you should know that during the G2 phase, the chromosomes exist as sister chromatids. At this point, the cell is really, really preparing to divide because that's the next stage, right? Well, this is when it leaves. Once it leaves the G2, it leaves interphase and it starts going into the mitotic phase. In the mitotic phase, the whole purpose is to divide the cell. So this is where it's really ramping up. It's getting ready to divide. So for example, some of the organelles are duplicated, right? Because again, we're going to take one cell and basically cut it in half to make two new cells. So we're going to need more organelles. If we're going to cut it in half, we want there to be enough organelles. Also, the cytoskeleton is dismantled. And we talked about that very, very quickly in that one. I think it was chapter 4. Still learning the new chapter numbers because to me, this is still a new textbook. But I forget the chapter number. But remember, we started the one about like how cells work. And I said, we're going to start this. We won't finish it. And I'm not going to test you on it. But some of this will come back later. So this is one of those things that came back later. The cytoskeleton. All you need to know, remember, that's just the thing that keeps the cell in this proper shape. And it's the thing that allows us to move things inside around the cell. But at this point, because the cell is about to be split into two, that is when the cytoskeleton is dismantled. So, any questions about G2. You guys are nice and easy. Finally, that brings us to the mitotic phase. So I'm going to be very clear here. We are now out, we are no longer in interphase, right? We're done talking about G1, we're done talking about S phase, we're now done talking about G2. So in the big picture, we are now on to the next big part. Now granted, when I say big, it's interesting because when you look at the little picture there, it's a very small sliver, which makes sense because like I said previously, interphase makes up usually about 90% of the entire cell cycle. So that means the mitotic phase, mathematically, makes up about 10%. So when I say big, I don't mean big as in time or chronologically. I mean big as in this is the important part of what we're talking about because this whole discussion is about cellular reproduction, and this is where cellular reproduction really happens. So this is where it gets a little bit complicated. No, not yet. It will get more complicated. This in itself would be nice and easy if we just left it at this, but so let's just start off. The mitotic phase itself is broken down into two overlapping processes. So again, remember interphase had those three stages, G1, then S, then G2. Mitotic phase is a little different. This is broken down into two processes, but again, like it says there, they overlap. So it's not like we go and start at one and then it stops and then we go into the other. It's more like we start at one, and as that first part is ending, the second part's already beginning. So it's overlapping, keep that in mind. Again, this is not a part of interphase. I'm going to circle that and make sure you are aware of that. The mitotic phase is the other part, so it's not a part of interphase. So when you get a question on the pre-lab that's due Friday, or today, for next week, there's a question that says something, I think, like list the stages of mitosis in order, I think it says. And then, or maybe that's a test question. I don't know. Anyway, if you're writing about the stages of mitosis, you should not write interphase. You will lose points for that because mitosis is a part of the mitotic phase. It has nothing to do, well, it's not a part of interphase. So anyway, the two overlapping processes, you should know this. This will definitely be a test question. Mitosis and cytokinesis. Those are the two parts of the mitotic phase. Again, they are overlapping, unlike the interphase, the parts of interphase. And to make things even more complicated, which really sucks for you, I'm sorry, mitosis itself is also broken down into different stages, and we're going to talk about that. That'll actually be the meat and potatoes of the exam. Most of the questions are going to be about the different stages of mitosis. We haven't even gotten into that yet. But yes, so mitosis and cytokinesis are the two overlapping parts of the mitotic cycle. Mitosis, you need to know this. This is what basically happens in mitosis, is the nucleus and its contents are divided into two daughter nuclei. Right? So if we have a cell, try to draw this. If we have a cell, oh, man, that's horrible, with one nucleus, if we were to just go through mitosis and not cytokinesis, what we would end up with is another cell, same cell, except now we would have two nuclei. So in a sense, the mitosis is when we duplicate the nucleus and everything inside of it. Specifically, and what we're going to focus on when we get there, when I say everything inside of it, what we're really focused on is the chromosomes. What happens to the chromosomes? In which stage do the chromosomes do this? In which stage do the chromosomes do that? So that's going to be the meat and potatoes of that portion of the exam. But again, mitosis is the part where we duplicate the nuclei, but that's not what we're after, right? We're after cellular reproduction. So duplicating the nuclei is not enough. And that brings us to cytokinesis. Cytokinesis is the part that actually splits the cell in half. So cytokinesis is the thing that gives us two different cells. So again, really quickly, if we were to go through, as you can see, that stupid drawing I have up there that I tried to make, if you were to go through mitosis but not cytokinesis, you would end up with one cell that has two nuclei, which doesn't really happen. Actually, you can look that up for independent work. How often does that happen? Likewise, if you were to go through cytokinesis but not mitosis, right? So if you had that cell split in half, then you would have two cells, one without a nucleus and one with a nucleus, right? Because if you do cytokinesis but didn't do mitosis, then you wouldn't split the nucleus into two. So if you only have one nucleus, then only one of the cells would get it. That's another thing you could look up for independent work. How often does that happen? Is there, or does it happen at all? Is there a name for it? Is there something that causes it? What happens to the body if that happens? Things like that. So anyway, the combination of the two produce two genetically identical daughter cells. And this is something we mentioned earlier, I think, Wednesday, when we first started this chapter. That's something you knew, and I'm bringing you back again, right? So the product of mitosis, and this is definitely a test question, this is definitely a lab question, it's very important. The product of mitosis is two genetically identical daughter cells. So the next word for attendance is, let's see, the mitotic phase is made up of two overlapping phases. One of them splits the nuclei. One of them splits the cell. Which of the two is the one that splits the nuclei? Nucleus, excuse me. It's not a nuclei until it gets split, because nuclei is plural, right? So which of those two up there on the board is the one that splits the nucleus? You guys, hey, you don't ask each other. You gotta figure it out.
No, I got it.
Okay. You just confirm them with each other?
Yep.
That's all right. Teamwork, right? All right. So you guys good?
Mhm.
All right. And remember, if you're watching the video, not live stream, but if you're watching the video, I know I didn't remind you earlier, but take a screenshot of this screen. All right. So that is it for the cell cycle in general. Now we can finally zoom in and focus on mitosis and cytokinesis, which again will be the majority of this portion of the exam. So when we talk about chapter 6, most of the questions are going to be about this next bullet point. Before we do that, let's do some reminders. So I remind you, pre-lab 7 is due today. I talked about it on Wednesday. I don't remember off the top of my head, but if I remember correctly, there was a video and there are pre-lab questions, but I don't think there are hypothesis questions. But there is a video, so make sure you do that. Prioritize that over lab 6, because again, well, you know what, I don't want to say that. Never mind. Also to remind you, lab 7 is completely virtual. So if you happen to get it done before next Wednesday, if you get it done and I have it graded, then you don't have to come in. And I'll let you know. Unless you get an email, unless I tell you don't have to come in, you should come in. But yes, there is a possibility you'll do it early enough and do it well enough where you don't have to come in. So that's it for pre-lab 7. Lab 6 is due today, technically. I always say this, let me help you, right? So let me help you do it if you haven't done it yet. We can sit down online or in person, whatever, and I can help you answer the questions and guide you through it. Or, this is really important, more so than any other lab you've ever done, if I've already graded it, come to me for feedback. Let me explain what you missed. So either A, you can fix it and help your grade, or B, you can get your 5% boost to your grade. But more importantly, because you have a lab report due, you really should come to me and let me give you feedback. Because if you don't understand what's going on, then how are you going to write a lab report about it, right? So even some of you, and this might blow some of your minds, because some of the grades have been pretty bad, that I've actually gone pretty easy with the grading. Like there's been a lot of small mistakes that I haven't even deducted for. So even if you got a decent grade, you should still come to me for feedback, because again, you have to write a lab report on it. And there's some stuff where you didn't lose points for it on the lab, but if you wrote that on the lab report, you would lose points. So at the very least, if you want, maybe come to me and get the feedback for the experiment that you did. Anyway, other things to remind you, if you did get a horrible grade, first of all, you can fix your mistakes to get some grade back, get some of your points back. And if you go to that spot on the course announcements under lab 6, you can do the virtual lab, right? So the virtual lab, you can apply those points to your actual lab. That also goes for the students who didn't come to lab on Wednesday. Remember, the best you can get is 1/4 credit. So if you want to make up for that, you can do the virtual lab in addition to the real lab and get some of those points back.
Question.
Yes, good.
For the lab report, is it only for the one you did or for the whole thing?
Good question. And I'll remind you, so the answer is only the one you did. And also I'll remind you, because I've said this and I'm not getting on to anything, so the questions are good. But if you ever have any questions and can't get in touch with me, remember, we had a pre-lab lecture where I explained that and other things. So if you obviously ask me, you did the better thing. But if you don't feel like asking me or you can't reach me, go back to that video that I've already posted and watch the pre-lab lecture. Because not only do I explain that, but I also explain how to write a lab report. So again, when you find yourself writing a lab report and you need help, or for some reason you don't want to come to me, or you can't come to me, watch that video. That explains everything. So the lab report is technically due 2 weeks after Wednesday, right? But 2 weeks after Wednesday, so that's not next week. That's not next Wednesday, but the Wednesday after that. And we're gonna be on spring break. So I don't think that's fair. So I just have extended it to the Monday after spring break. Because Monday will be our first day back. So any other good questions about lab report? Any other questions?
Lab report for the lab we just did is after school.
Yeah, technically it should be due the Wednesday of spring break, but that's not fair, so I'm doing it Monday after spring break. Just as usual, like everything else, please come to me. Oh my God, it would save me so much more time and give you guys so much better grades. Before you submit something, meet me online, at least just for the big stuff. Like, hey, will this even give me any credit? And I can look at it and say, whoa, that's 0 credit. You need to do this big thing first. It would be so quick. Please let me help you. Someone's already turned in a lab report. Awesome. But to remind you, you need to follow the directions that are in the lab report section of My Online, right? You need to use the form that I gave you that has all the little bullet points. And then under that, that's where you write your lab report. And you're going to save it as a PDF and then upload that. But yeah, let me help you. Please, please, please let me help you. So anyway, any questions about the lab report or lab 6? All right, exam 2, like I said, technically it was supposed to be later, but we're a little bit ahead of schedule. So exam 2 is already open. I opened it early. And because it's open so early, this will be due the Sunday of spring break. In theory, you should get it done before you get to spring break, but it is spring break, so who cares, right? I'll just extend the due date. So it is due on the last Sunday of spring break. As usual, as always, it's about 50 questions to answer. About 50 minutes to answer 50 questions. If you want more time, we can meet online. If I can see you and see your screen and hear you and know that you're taking a closed book exam, then you can take essentially as long as you want. Any questions about exam 2? All right. Also, when exam 2 closes, so does the retake opportunity for exam 1. So I'll remind you, you can still retake exam 1. It's open book, it's not timed, you can take it as many times as you want. The only catch is you have to tell me when you're done. So you go, take it as many times as you want, and then say, okay, I'm ready for you to adjust my grade. And then I'll go in and look, and I'll average your first grade with your highest grade. So even if you take it 10 times, and your highest grade was the 3rd time you took it, then that's the one I'm gonna take, right? So I'm gonna average your first grade, and then in that case, that example, the 3rd time. So far, I don't think anyone's taken me up on this. I don't know why. You have nothing to lose. When you take your final exam, it's going to be based on your first 3 exams. So this is also going to help you prepare for the final exam. You have nothing to lose. If I were you, I would retake it. And I know this sounds a little bit harsh, but first of all, I don't adjust grades at the end of the semester anyway. So I wouldn't waste your time asking me that. But really don't ask me that if you haven't taken up these opportunities. I'm only 5 points away from passing. Is there anything I can do? Yeah, you get a time machine and go back and take the first exam like I've been begging you to. So anyway, that's it for announcements and reminders. Do you guys have any questions or your own announcements before we move forward? All right, let's jump into mitosis. So remember, all of the cell cycle is broken down into interphase and the mitotic phase. We've learned that. Then we said all of the mitotic phase is broken down into mitosis and cytokinesis. We know that. Now we're saying the mitotic phase itself is broken down into 4 distinct phases. So not overlapping, but 4 distinct phases. And I don't remember why I had a star there, but let's hope it wasn't important. Oh yeah, the star is this. Okay, so if you look at your textbook is actually, I'm giving you the Cliff Notes version. So the textbook doesn't say prophase, metaphase, anaphase, telophase. They throw in another phase. So I'm giving you the easier version. I'm going to test you off of what I'm giving you, not what the textbook's saying. So for us, there's only 4 phases: prophase, metaphase, anaphase, telophase. I suggest that's the first thing you memorize. Know them in order. There'll be 2 test questions that are going to look very similar, and don't confuse them. One of them is going to say, remember this is multiple choice, which of the following
are the mitotic phases in the proper order, right? And only one of them, one of them will say prophase, metaphase, anaphase, telophase. That'll be the correct answer. Then there's going to be another one that says in blank phase, the chromosomes do this. In blank phase, the chromosomes do that. In blank phase, the chromosomes do this. And in blank phase, the chromosomes do that. That's going to be different. That's not the answer to that one's not going to be prophase, metaphase, anaphase, telophase. That's me asking you what happens in the different phases. So don't confuse the two. Someone always does. Anyway, yeah. Each phase, again, this is what we're going to focus on mostly. There are some exceptions, but mostly what these questions are going to be about, like I said earlier, for the most part, it's all about the chromosomes. So again, I'm going to say which phase do the chromosomes do this, and which phase do the chromosomes do that. Now, granted, some other stuff is happening during prophase and telophase, and I'll tell you what they are when we get there. But for the most part, it's going to be about the chromosomes. And again, we're about to discuss each of those separately. So again, prophase, metaphase, anaphase, telophase. Memorize that in that order. And then, when you're studying, then once you have that memorized in that order, then start memorizing what happens. And when I teach this, I'm hoping this is actually pretty logical. I have a feeling, and I'm going to try to get you to answer these questions. I'm going to see if you can guess. Like I'm going to tell you what happens in the first phase, and then before I tell you what happens in the second phase, I'm going to see if I can get you to guess. And hopefully you will, because hopefully this is very logical. Any questions about this slide? All right. First of all, before we can talk about what happens during this whole thing, we need to talk about something called the mitotic spindle. Not that I'm probably going to ask you a question about it, but I'm definitely going to use this term. And really, this whole thing happens because of the mitotic spindle. The mitotic spindle is a football-shaped structure. It consists of microtubules, which is one of those things we learned about in that chapter that we flew through. And I said, I'm not going to test you on that. The important stuff will come back up later. Here it is, right? So we've got these microtubules, which as far as we're concerned, are ropes or strings. It doesn't matter, right? What they are things that attach to the chromosomes and literally move the chromosomes around. So the mitotic spindle is the thing that has, the microtubules. And the microtubules are the things that attach to the chromosomes and move them around. And remember, as far as we're concerned, in this conversation, that's mostly what we're concerned about, is what happens to the chromosomes. So, and where do they go? So, obviously, this is very important for that conversation, because it's the mitotic spindle that's moving the chromosomes. All right, any questions about that slide? Very good. Let me ask you this. All right, if we had a race, it's kind of hard for you guys to see. It might be easier online. What we have here on the right is 1, 2, 3, so 9 balls of yarn, right? What we have on the left is also 9 balls of yarn. Now let's say I had you guys race, and I gave you these 9 balls of yarn. You get the ones on the left. I gave you the ones on the right, and you guys have that whole table to yourself, right? And I say, all right, I want you guys to separate it. It would be better if it was an even number, but let's assume it's 10 balls, right? I need you to have 5 balls on this side, 5 balls on this side. Who do you think is going to win that race? The person who has these balls on the left or these balls on the right? On the right. And why is that? Because they do that.
You just set them up, pull them up.
Yeah.
You gotta untangle them.
Exactly. On the right, you've got like individual little balls. Okay, just move them. Here on the left, you got a big squiggly mess, right? So think about the chromosomes. Chromosomes are the same way. That's the purpose, right? I mean, it's not necessarily a race, but we're trying to separate the chromosomes to different sides of the cell, right? Because we're about to split the cell. So think about that, and hopefully logically this makes sense. One of the first things that happens, all right, let's jump into it now. We're talking about prophase, the first phase. One of the things that happens in prophase is the chromosomes coil up. So you go from a big squiggly ball, squiggly mess of yarn to balls of yarn, so to speak. So now, that way they can be moved. So that's one of the things you need to know for the exam. In prophase, the chromosomes coil up. And again, I'm hoping that's very logical, intuitive. Because if you're going to separate them, it would be a lot easier if they were coiled up. As you know, because we already talked about this from S phase of interphase at this point, Each of the chromosomes exists technically as sister chromatids that are joined at the centromere, right? Because they've already duplicated themselves. That's not necessarily going to be a test question. This is why it's not in bold. So it's just a reminder to remind you where we're at here. But yeah, the chromosomes are coiled. Technically, they're sister chromatids at this point. Also, imagine too, going back to this, you guys are in the race. You guys both, you've got the ones on the left, you've got the ones on the right, but for both of you guys, I have the balls of yarn in a bag, right? So you get a bag of yarn, and you get a bag of yarn. And I say, all right, I need you to move them to separate sides of the table. What's the first thing you're gonna do?
Take it out of the bag.
Take it out of the bag, right? Same thing here. If we're gonna move chromosomes from one side of the cell to the other, we've got to take them out of the bag. In this case, the bag is the nucleus, right? The nucleus is containing them, so we need to get rid of it. So in prophase, the nuclear envelope breaks into pieces. As far as we're concerned, it disappears. I put a star next to this, I think, to remind myself that your textbook might get into more detail. If it does, don't worry about it. I'm not going to get into more detail for the exam. What you need to know for the exam is during the prophase, the nuclear envelope breaks into pieces. So for example, one of the questions might be, in which of the 4 phases does the nuclear envelope break into pieces? And the answer is prophase. In which of the 4 phases do the chromosomes coil? Prophase. All right. So again, the whole idea here is to move around the chromosomes. So if we're going to move around the chromosomes, then we need to attach the things that move them. And that's this bullet point right here. The spindle attaches to the centromeres. I don't remember why I have these stars. Again, I'm assuming it's because the textbook goes into more detail. But for you, that's what you need to know. The test question might be, in which stage of the 4 stages does the spindle attach to the centromere? And the answer is prophase. And again, this should all be very logical. If this is the first phase, all these things have to happen before we can move around the chromosomes. Now, once all these 3 things happen, finally, we can start moving the chromosomes toward the cell, center of the cell. So prophase is defined by all these things. You'll need to know that for the exam. And again, during prophase, once all these other things happen, the chromosomes are moving towards the center of the cell. So let's see if anyone can guess. If that's the last thing, that's my last description of prophase, is the chromosomes are moving towards the center of the cell. Then what do you think the next phase is going to be? Like, where do you think the chromosomes are going to be for the next phase? What defines the next phase as far as the chromosomes position? Because here they're moving towards the center of the cell. So what do you think the next stage, where do you think they'll be in the next stage? No guesses?
Outside of the cell?
I like that. I like that too, you guys.
Lined up in the corners of it, so we can read that.
Oh, in prophase, they're all over the place in prophase, but they're starting to move wherever they are. They're starting to move towards the middle. So where do you think they are in the next phase? There you go, in the middle, right? So that's very logical, right? If I'm telling you here that in prophase they're moving towards the middle, then the next stage they are in the middle. But anyway, before I show you, here's some pictures of what we're talking about. This is not from your textbook. This is from the old textbook. You can see this is interphase right here. So we haven't even gotten into mitotic phase. You can see the big squiggly mess of chromosomes. The centrosomes have formed, like I mentioned earlier. The nuclear envelope is still there. The plasma membrane, all that's still there. Now finally we get into prophase, which is what we just got finished talking about. The mitotic spindle starts to form. It attaches to the chromosomes, as you can see here. The nuclear envelope has disappeared, which you can't see because this is not animated, but these chromosomes are starting to move towards the middle of the cell. And that brings us to the next phase. Like you guys already said, that's when they're in the middle of the cell. There's a lot of words on this slide, but for the exam, that's all you really need to remember. Metaphase is when the chromosomes are in the middle of the cell. It's when they met in the middle. That's how I always remembered it. Metaphase is when they met in the middle. Anyway, the way your book describes it, it's when the centromeres line up between the 2 poles of the spindle. And that is something we call the metaphase plate. We'll put a big old X through that, not because it's not true, but because I'm not going to use that verbiage in the exam. I'm not going to get that complicated. The point is that the chromosomes are lined up in the middle of the cell. That's what you need to know for the exam. That's it for metaphase, as far as I'm concerned. In metaphase, the chromosomes are lined up in the middle of the cell. Now, mind you, they're still attached to these centromeres, which are moving them around. And at this point, since they're lined up in the middle of the cell, what's happening is it's like a tug of war going on. And the tug of war is they're trying to pull those sister chromatids apart. Right? In metaphase, they're in the middle, and they're trying to get pulled apart. So, if in metaphase, the sister chromatids are trying to get pulled apart, what do you think is going to happen in the next phase? They do get pulled apart, right? This is all very logical, yes. Anyway, there's a picture of metaphase. You can see all these chromosomes lined up in the middle. So in the next phase, yes, they're pulled apart. The sister chromatids are separated. At this point, technically, they're just full-fledged chromosomes now, right? Because now they're not sister chromatids, they've been separated. But yeah, as far as the exam is concerned, that's what you need to know. The sister chromatids are now separated. Because remember, in the last part, they were getting pulled apart, or they were getting pulled in that direction. But as soon as that happens, as soon as they get pulled apart, that is when it's considered anaphase. And again, that first bullet point, that's what you need to know for the exam. They are separated. But let's look at these last two bullet points to help you guess the next stage. So in anaphase, again, they've been pulled apart. Now they're moving towards the opposite poles of the cell, opposite sides of the cell. So, given this bullet point right here, since they're moving towards opposite poles of the cell, what do you think, where do you think they're going to be in the next phase? If in anaphase, they're moving towards the opposite poles of the cell, where do you think, by definition, they will be in the next phase?
Opposite poles of the cell.
Opposite poles of the cell. You guys are great. Exactly. And that's it. Well, that's not it. In a sense, I mean, you're correct. Yes, they are now at opposite poles of the cell, but telophase, at least academically, as far as the exam is concerned, is basically like the opposite of prophase, which should make sense. You've already accomplished what you needed to. You separated the chromosomes. So now you can create your nucleus again, right? That's what that bullet point is. The nuclear envelope reforms. There's no longer any need for them to be all coiled up, so they uncoil, right? There's no need to move them around anymore, so the mitotic spindle disappears. And again, I don't remember what this little star is for, but I assume it's because your textbook goes into more detail. So at that point, mitosis is finished. Not the mitotic phase, because remember, I'll remind you, the mitotic phase is broken down into mitosis and cytokinesis. But mitosis itself is complete. And depending on your source, some sources say that cytokinesis, speaking of which, remember I said these are overlapping phases, right? The mitotic phase and cytokinesis. So according to this textbook, telophase technically occurs cytokinesis usually starts during telophase. Other sources might say something different, so I'm not going to ask you that. So again, as a reminder, I'm going to try to visualize this for you. It's that phase, and then there's this phase. So it'll be better if it was different colors. So they're overlapping, right? So if this is the mitotic phase, from here to here, this might be cytokinesis, from here to here. They overlap. And because the source is different, I'm not going to ask you technically when cytokinesis starts. Your textbook tells you, but I'm not going to ask you. Some other things to consider for independent work. I said here that the spindles disappear. Well, nothing really disappears. Academically, for the exam, we can consider them disappeared. But for independent work, you could ask yourself, where do they actually go? They don't disappear. What happens to them? What is the thing that breaks them down? Like what cellular apparatus does it? What is the name of that process? All those questions you could ask yourself. Let's see. This one's going to be an easy one, the next word for attendance. And again, if you're watching the video, not if you're online, but if you're watching the video at a later date, you need to take a screenshot of this slide. What happens to the nuclear envelope in telophase? Does it disappear or does it form? I'll give you a multiple choice question. In telophase, which is the one we're talking about now, not prophase, which we talked about earlier, does the nuclear envelope form or does it disappear? All right. Any questions about telophase or mitosis in general? Because now, if not, we're going to talk about cytokinesis. Anyway, there's a picture of the telophase. It's an actual electron microscope picture. All right, cytokinesis. Again, this is where the cell actually separates. We've already separated in mitosis. We separated the chromosomes. We turned one nucleus into two nuclei. Now we need to do the same thing for the cell. We need to turn one cell into two different cells. Before we talk about that, your books point out that it's different in plants versus animal cells. And again, I'm going to put it next to this because I'm not going to ask you. Usually, cytokinesis starts, like so splitting of the cell, usually starts, in animal cells anyway, during anaphase. So while those, which kind of makes sense, right? While those chromosomes are getting pulled apart, which is what anaphase is, right? They're pulled apart, they're going towards opposite ends of the cell. Not only that, but the cell itself is getting elongated, right? Not that I'm going to ask you that, but there you go. Now you know. So again, the most important thing you need to know for the exam is this last bullet point right here. What is cytokinesis? It's when we divide the cytoplasm. It's when we turn one cell into two cells. And again, this top bullet point, I might ask a couple of questions about it, probably not, but I will give you this information on the upcoming slides. But before I do, are there any questions about this slide? Okay, in animal cells, this is known as cleavage. You can see why. Look at that picture. It looks like a wrinkly old Pac-Man. You guys might be too young to really remember what Pac-Man looks like. But anyway, yeah, in animal cells, we call this process cleavage. And it begins with the appearance of this cleavage furrow that you can see right there. It happens right there in the middle of the cell. I'm not going to ask you any of those questions. If anything, and I'm probably not going to ask you this, but if anything, maybe be aware that it's called cleavage, just in case I use that word in the exam, but probably not. There's a different picture, right? So here you just have you can see this big blob, this one cell that has the two nuclei, and it's starting to move inwards until we have two new daughter cells. That's it. Nice and simple. Plant cells, if anything, you just need to know it's not that simple. Because remember, one of the things you definitely need to know is that plant cells, have cell walls and animal cells don't. Basically, I won't even talk about the rest of this because that's basically all you need to know. Is that during cytokinesis, for plant cells, they also have to deal with the cell wall. So they can't just pinch off two new cells. Instead, what happens is they build a wall in the middle of the original cell, and now we have two new cells. That picture kind of visualizes it. This picture is not from your textbook, I don't think. So again, in your mind, you can just think in an animal cell, you have animal cytokinesis kind of squeezes off and pinches until you get two new cells. In plant cells, you're just building a wall in between. It's almost like, never mind. You're just building a wall in between the two cells. And once that wall is completely formed, you then now have two new cells. And that's it. Any questions about that? You can see there's a bunch of words here like vesicles and cell plate, things like that. And your textbook goes into great detail about it, but I'm not going to ask you any of that stuff. Unless it's for extra credit. And right now, I don't plan on having any extra credit on the exam, since you'll be able to retake it. Anyway, that's it for the entire cell cycle. We've talked about interphase and its three parts. We talked about the mitotic phase, which is broken down into overlapping mitosis and cytokinesis. We talked about that. Now, the next thing we're going to talk about is there are these exceptions to the rule. Not all cells follow the classic cell cycle. And rather than start that, let's not start that. Let's see. Yeah, so that's it. So the last word for attendance, again, send me a question. Question about something the material that I presented. So, I mean, you can send me any question you want, but as far as the last word for attendance, the question needs to be about this material. And that's it. We'll end 5 minutes early. I'll be online hopefully all day. For the most part, I've got some meetings to go to, but for the most part, I'll be online. If you need some help with Lab 6 or pre-Lab 7,